Well, if bacteria are living things and some of them are heterotrophs, meaning they have to eat other things, what is it that they eat? And do they break that down like we break down our food? Well, do bacteria eat sugars? Actually, this right here is lactose. That's a sugar that we find in milk. And yes, there are some bacteria, some lactobacillus, that actually break down lactose and eat it. How does that process work? What's the digestion like for a bacteria? How is that different than our digestion? That's what we're going to look at in this video when we talk about how bacteria use their DNA, their genes, and the expression of those genes to break down food to make different amino acids. Let's get started. So if you've been watching my video series, you probably remember when we talked about metabolic pathways. So when we talk about metabolic pathways, we said that in one case, we're going to take this substance, this chemical here, and through a series of pathways, convert it to that chemical there. So that process of converting this chemical to this chemical to this chemical to this chemical and ultimately this chemical is done by a series of enzymes, right? So enzyme 1, in this case, takes chemical A and turns it into B. Enzyme 2, B to C, et cetera, down the pathway. Now, you may also then remember we talked about feedback inhibition. This is when we get enough of chemical E. So let's say that we're taking chemical A through a series of steps, breaking it down to chemical E. Well, what happens in the body when we get enough chemical E? We don't need to convert chemical A to chemical E anymore when we already have enough of chemical E. So you may remember when we talked about enzymes, we talked about sometimes chemical E, or whatever chemicals at the end, will bind to the first enzyme allosterically and inhibit it, change its shape so that A no longer fits in the process. So another way, this is a way to control whether or not chemical metabolism in the body is occurring by taking one product and using it to block a pathway. Okay, so we've talked about that in previous videos. Well, there's actually a different way that organisms can stop this process, right? If we don't need chemical A to be converted into chemical E, we could use the enzyme method like we saw here, or we can actually use the genetic control method. And that's what I want to talk about now. So here's another way to regulate metabolism. We do it through the gene itself. So what you've got to remember is, if you think back to these enzymes, right? This enzyme converted A to B. This enzyme converts B to C. These enzymes are crucial in this role. Well, one way to stop this process from happening is to stop the enzymes from even being made. Well, how are enzymes made? Well, they're proteins, right? And we know that proteins, we learned in our last video, are made by the transcription and translation of DNA. So gene 1 in our DNA will ultimately code for enzyme 1, the protein, to be made. So if I don't want any of E to occur, chemical E, all right, I don't want this process to take place at all, what we can essentially do is block the gene from being made. So in some cases, chemical E can actually go down and block the transcription of the gene so it's not even made into an enzyme. That then, because we don't have the enzymes, stops the whole process. All right? That's really what this video is about. Regulating metabolism, regulating things through genetic expression. All right, so let me give you a few examples. In this video, we're going to focus on bacteria because it's a little more simple than focusing on gene regulation in us. In my next video, we'll talk about how gene regulation occurs in eukaryotes, but in this video, we're going to specifically talk about bacteria. All right, so how does this work? Control of gene expression, right, whether or not this certain gene is expressed, enables individual bacteria to adjust their metabolism to environmental changes. So, let's take a look. Here's a bacteria, and if we want to regulate the gene expression of that bacteria, we basically either need to turn a gene on or turn a gene off. In other words, if the genes are normally turned off, we might want to turn them on. Conversely, if genes are normally sort of in their on position, well, we might want to turn it off. How do we do that? We do that through things called inducers and repressors. Basically, these are small molecules that interact with some proteins to either start or stop transcription. So let me give you an example down here. In this case, right, here's some DNA, right? So this is DNA right here. And RNA polymerase, remember, that's the enzyme that's going to do transcription. It's ready to go. But, oh, look right here. Look, we got a repressor. This repressor is bound so that RNA polymerase cannot come and transcribe these genes, okay? So a repressor, in essence, makes no transcription. So it stops transcription and thus translation, making the protein from happening. Okay, well, what about inducers? Well, if a repressor represses and stops, the inducer might remove the repressor. Okay, so if we normally have a repressor on, if we have a chemical, in this case allolactose, binds to the repressor and removes the repressor. So in this case, allolactose is called an inducer because what do we see? We see an open area here on the DNA so that RNA polymerase can now come and do its transcription. All right, so this was just an introduction. I'm going to go into each of these types in more detail. All right, before we can really dive deep, we need to understand that sometimes in bacteria, genes are grouped together. So let me give you an example. An example of this genes grouping is called an operon. An operon is when genes are grouped together that have related functions. So remember that slide early on where we talked about all those enzymes in a pathway that, A goes to B, goes to C? Well, those genes that are grouped together would code for those enzymes. So it makes sense that they're close to each other on the DNA. Now, on an operon, you've got a few regions. All right, so here's our DNA. Now, I'm going to put an overlay on it, but it's still going to be DNA. It's just going to show you the different regions of the DNA, okay? So here are some regions we want to talk about. This right here is an operon. The whole thing is an operon, right? Genes grouped together. Right here's gene 1, gene 2, gene 3, gene 4, gene 5. All of these genes right here are grouped together because they're going to be involved in a similar function. Now you remember RNA polymerase. RNA polymerase is what is going to transcribe these genes into messenger RNA. Then that messenger RNA, of course, is going to go out and find a ribosome. Boy, that's bad. A ribosome to translate it into a protein. Okay? So that's the ultimate goal, is that we're going to get a protein, an enzyme made. But what are the parts? So here are the genes, these 5 genes. Before that is something called an operator, and before that is something called a promoter. Let's see what those are. A promoter is the place that RNA polymerase binds. So RNA polymerase, the enzyme, is going to come, it's going to bind to this promoter region, and then it's going to transcribe this gene into RNA, which eventually will go into proteins. The operator is where the regulatory proteins often bind. These regulatory proteins, we talked about repressors, and then sometimes inducers that will start this transcription process. So an operon basically is just this group of genes similarly related where there's a promoter and an operator. Now, as I mentioned, if this transcription occurs, these genes will be transcribed into messenger RNA, And then ultimately, those genes will be translated by the ribosome into these enzymes or proteins. Okay? So, let's start with how a gene can be turned off. So, let's take you back. Remember when we were talking about the very beginning how chemical A goes to B, goes to C, whatever, ultimately goes to E. Let's say in this case, chemical E is tryptophan, which is a nice amino acid that bacteria may need to make. And what you see here is an example of tryptophan, all the genes, all the enzymes that are going to code for the enzymes to make tryptophan. All right, so first step in protein production is transcription. So if we stop this transcription, we've turned the gene off. Well, how do we stop transcription? Obviously, we need the repressor. So, how does the repressor know to go and bind to the DNA? Well, this repressor protein will bind to the DNA, blocking RNA polymerase from transcribing it, when it has a certain specific signal to do so. What's the signal? Well, in this case, what we're seeing, tryptophan itself is the signal. In other words, why would a bacteria need to make all these genes or transcribe all these genes into all these enzymes to make tryptophan if I already have, or the bacteria already has, enough tryptophan? We don't need to make more tryptophan if we've got enough. So tryptophan actually acts as a co-repressor, binds to the operator, so we stop transcription. Okay, so I'm going to go over this specific in a little more detail here. So tryptophan is a repressible operon. Normally notice what RNA polymerase is doing. RNA polymerase moving down the DNA, making this nice messenger RNA for us, right? Here's the operon to make all these tryptophan enzymes. Well, what happens when we have enough of this done? We've made all the tryptophan we need. Well, in this case, tryptophan will bind to a repressor, activating that repressor so that the repressor, as you saw here, comes and binds to the operator site. Here's our promoter region, here's our operator region. The repressor's bound here. So can RNA polymerase come in? No, it cannot. So RNA polymerase cannot bind, so it has to go on. So this is a way that we turn a gene off. This is what we call a repressible operon. Tryptophan is an example. Why? Because tryptophan serves as the co-repressor, which then activates this spot, activates the repressor, binds to the operator region so that RNA polymerase cannot transcribe these genes. And if they don't transcribe the genes, then we don't get the enzymes to make tryptophan. Okay? So, that was a reversible operon. An important thing to note, how does tryptophan operate the repressor? Well, it does it by binding to the repressor and resulting in a conformational change, right? So it conforms the shape so that it no longer fits and can bind to that DNA. Therefore, RNA polymerase would not be able to do its job. Okay. So, what's the opposite? How do we turn a gene on? So, regulatory proteins stimulate gene expression by binding to DNA and stimulating transcription. So, let me give you an example. So, in terms of positive control, you remember the normal situation. In this case, we're going to talk about lactose, how I started the whole video. If a bacteria, for example, a bacteria wants to digest lactose, right? So it's got some milk sugar and it wants to break it down. Well, how do bacteria break down lactose? Oh, they have a bunch of enzymes that will take lactose and break it down. Okay, well, where do they get those enzymes? Well, it's in the bacteria DNA. How do we get those enzymes made so that the enzymes can break down lactose? Well, we actually need to transcribe them first, right? So here's our lactose operon. You see the 3 genes here that are going to break down lactose. Right now, this is not being transcribed. Why? Oh, there's a repressor in its place. Well, when lactose is present, right? Lactose is detected. So the bacteria is taking in some lactose. It's getting that milk sugar. That lactose actually binds to the repressor, making a conformational change, so it is now removed. Remember in the previous case, the tryptophan bound to the repressor to make it go to the DNA. In this case, it's the opposite. Lactose is binding to the repressor to get it to remove itself from the DNA. All right? So I'm going to go into a little more detail on this specific one so that you hopefully will understand it a bit more. Because once this repressor is removed, now notice RNA polymerase can do its job of transcribing these genes into the enzymes which will break down lactose. Okay? Lactose. So let me show you this again. This is normally, so this operon is normally off, right? The repressor is bound. We're normally off, right? Because basically, if I'm a bacteria, I don't need to make a bunch of enzymes to digest lactose if there's not any lactose to digest. So when the bacteria takes in some lactose, all of a sudden, oh, it now needs to make the enzymes to break down that lactose. All right, so we're normally in the off position. We have the repressor protein. So if RNA polymerase comes in, it can't come in, it can't do its job, so it just has to go along with its merry way. However, if the bacteria has taken some lactose, and this is just a symbol. We know lactose is actually a disaccharide, so there could be another little sugar on there. But for simplicity, I'm just showing it as one here. So when lactose is present, it actually goes and binds to the repressor protein. There it goes, binding. Now what can happen? That removes it. So lactose binding to the repressor protein actually removed it so that RNA polymerase can transcribe these genes. What's it transcribing them into? What's transcribing it into messenger RNA? That messenger RNA will then go and be translated by a ribosome into the enzymes that will break down lactose. Okay? All right, so complicated, yes. This can be a very complicated idea, but we're talking about bacteria, and we're talking about basically the stuff that we know. Lactose serves as a co-repressor to remove it. In this case, because it's inducible, we turn it on. Tryptophan, what we saw before, actually is a repressible operon that represses the transcription. So hopefully in this video, this helped you understand a little bit more about gene regulation. All of what we talked about here was in bacteria. In my next video on eukaryotic gene expression, we'll get a little more in depth about how we actually regulate our DNA.